So maybe we should start. So it is my pleasure to introduce Dr. Jeff Conner. He's a distinguished professor in evolutionary biologist. He completed his bachelor's at Harvard University and his PhD at Cornell. He's currently the interim president of the Kellogg Biological Station and a professor in Michigan State University. He has published many peer-reviewed papers, and he is the co-author of the book A Primer of Ecological Genetics. I highly recommend. And his lab studies how natural and sexual selection in plants leads to adaptation to variable environments, while also exploring potential constraints on these adaptations. In addition to his research, Dr. Conner has been actively involved in academic service, including serving as an editor in several journals, and he currently holds the presidential position of the American Society of Naturalists. He's also a dedicated mentor and have received multiple awards for this contribution to mentoring. And today he will be talking about some work in his lab about the mechanisms behind the loss of traits that have lost its functions, specifically in the two short stamens in Arabidopsis. Please join me in welcome Dr. Conner.
Thanks, Juan Diego. You can hear me well? Okay, good. I appreciate all those nice things that you said. I also appreciate things you could have said and didn't. You could have referred to me quite accurately as a well-meaning elderly man with a poor memory, but he knew that might be inappropriate. I think I mentioned, maybe at breakfast or something, I realized last week that I spent the summer here at Purdue 50 years ago. 1974, I had just been promoted to full professor. I'm not that old. No, I was in high school, and it was a high school program for biology nerds, and it was I came from a small town in upstate New York, and it was a pretty transformative experience for me. I spent time in Lilly Hall. I recognized it a little bit. It was a long time ago. Anyway, so today I'm going to talk about some of the work in my lab, as Juan Diego said, and there's the title that I provided, so I kept that there, but I'm actually going to talk more generally about stamens in the Brassicaceae and our work in wild radish, which is a quite different system. So first I have this slide, and this looks like maybe the standard. You've seen other biodiversity slides. You know, I study biodiversity, and that's not, well, it is that, but this is here to talk about adaptations, because that's really what I'm interested in, is phenotypic adaptations. And so I have some really great examples here. The jellyfish with, I don't even know, tentacles and the mantle and all those things that jellyfish have. And here's a cuttlefish with tentacles and eyes that are similar convergently to vertebrate eyes, and they change color. And here's the duck-billed platypus, which is just crazy. But all these adaptations that a mammal probably shouldn't have. This is one of the orchids that I think really does look like a bee and tricks stupid male bees into mating with it and getting pollination done, and the bees don't get any. Uh-oh. Yes. Keep going. That's what they said. Especially if we can bring them down in front specifically is good, but anyway, and these are the fungus beetles that I actually did my thesis work on. Is that okay? Everybody's going to fall asleep. Yep. I'll talk really loud. There's still these guys are nocturnal, so it was better before when the lights were out. Anyway, the males have horns that they use to fight for the females in the fungus beetles. So lots of adaptations, and so why study adaptation? There's Chuck there, and here's what he said: that perfection of structure and co-adaptation which most justly excites our admiration. So I don't talk like he does. I say this. I think a lot of us got into biology because of all the cool things that organisms do. Those are adaptations. And all life on Earth depends on adaptation, right? Without adaptation, species go extinct. It's not perfect like Chuck thought. Adaptation certainly isn't, and it certainly isn't a trivial problem because almost all species that have ever existed on Earth have gone extinct, which at some level is a failure of adaptation. So two things that can cause failure of adaptation perhaps are genetic correlations. We think primarily caused by pleiotropy, right? Single genes affecting multiple traits, and possibly genetic drift. And so these are the kind of the themes that I'll talk about today. This is a plant group, but I still want to talk about Brassicaceae, mustards. I mean, I have these pictures that I took of Brassicaceae flowers because some botany folks think Brassicaceae have really boring flowers, 4 petals and a cross, 4 long and 2 short stamens. That it's definitely, that part's not boring. But there's more variation than people often think. Almost 4,000 species, it's a big family. It has all kind of crops, lots of vegetables that people don't like to eat very much. Although a lot of them, Brussels sprouts, right, with roasting, they've made a comeback. And of course, is a major invasive garlic mustard and the model organism that many people in this room study. So it's an important, and also agricultural weeds. And so that's what I'll talk about, some work on weedy radish. We are working on, in the lab, adaptation of weedy radish to agricultural fields. But today I'm just going to talk about flower evolution, which seems to not really even be part of the weed evolution story. Okay, so wild radish, if you're not, I call it a model system in herbivorous pollination. Right now, it's mostly my lab, but in the past, a lot of people have looked at plant-insect interactions, a lot of work on pollination and herbivory. It's an agricultural weed on every continent except Antarctica. Crop ancestor? Oh, no, well, I guess the native species that the weed, okay, so this says wild radish, okay? So the weed is Raphanus raphanistrum, but there are native, obviously, not obviously, there are native Raphanus raphanistrum, which may be the crop ancestor, but there's also a little known Raphanus pugianiformis that's endemic to the Eastern Mediterranean that I think may actually be the crop ancestor, and we're doing some work on that too, that I won't talk about today. So this slide's a little old. We've done a lot of sequencing and transcriptomics, and we're on our 4th reference genome that I think at least some of the chromosomes will be chromosomal scale. It's a self-incompatible annual, and so that's why I've spent since 1987 studying floral adaptation to pollinators in radish. And we're still doing that. It's a little bit more on the back burner right now. So one of the traits that I've spent a ridiculous number of years studying is anther exertion. So we define this, and I should have put it right on the slide, but so there's a tubular part of the corolla here formed by the claw, the inner part of the petal. It's not a true fused tube, but it functions as a tube in terms of pollination. So if we take the tube length and subtract, and I'm going to focus on the long stamen now, subtract the long stamen filament, right? Just this long thin part, not the anther. If we subtract filament from tube, that gives us the anther exertion. So this plant is slightly positively exerted. The bottom of the anther is just out in the open part of the corolla tube. And so what do we know about anther exertion? We know that there's stabilizing selection on it, but just by the bees. So my former student, Heather Solly, on the faculty at Shippensburg University now, showed that, looked at pollination by a number of different pollinators. 14 of the 15 genera are effective pollinators that Heather looked at. The 15th might be too, it just didn't show up in her study. But it's really only the small bees, which are one of the major pollinators, that exerted this stabilizing selection. We've done a bunch of functional studies looking at how anther exertion affects pollen removal, and again, the bees remove the most pollen in single visit with this intermediate anther exertion which is, I didn't really say, the stabilizing selection means you have the highest male fitness if you put the anther right here at the opening. If you put it up higher or down lower, you have lower lifetime male seed siring success, which we did by doing genetic marker studies. And the functional studies match that. We know that there's a really high correlation between the filament and corolla tube that's caused by pleiotropy or extremely tight linkage. And we did artificial selection, though, and were able to get rapid response and produce anther exertions that were not found in nature, that anthers that were way up here and some that were inserted almost completely into the tube. And what a study that we've had ongoing for a while is QTL mapping on crosses of those artificial selection lines to try to look at the genes underlying just that rapid evolution that we caused by artificial selection. So that's, I don't think more about anther exertion. Tetradynamy, which just means 4-2, and we often call it the 4+2 anther arrangement, which is diagnostic in the Brassicaceae, although there's a few genera that have secondarily lost it and have the ancestral condition of 6 stamens of equal length. And so I just said this, almost all the 4,000 species have this trait, but we don't know why. And my lab's been working on it for a while, and we know some things, but we don't know really functionally what the 2 stamen heights are doing. So let me tell you a little bit, I'm not gonna just quickly summarize the functional studies we've done. So short stamens produce more pollen per anther than the long stamens do, but less pollen is removed on each visit. And that's because they are inserted down into the tube and they just don't, the corolla tube, they just don't contact the pollinators as well. And by the way, this more pollen in short stamens is also true in Arabidopsis thaliana, and it really doesn't make sense there as well, it probably does make sense, but we don't know why it makes sense. Well, here's a picture of this. So here's a cabbage butterfly, which is a pest and a pollinator. And you can see that the body of the cabbage butterfly, the thorax is being contacted by the long stamens, anthers, but not by, there's a short stamen anther right in there. So it's just being missed by the pollinator. If we experimentally remove either the 2 shorts and leave 4 long, or 2 of the longs and leave 2 and 2, that latter has less pollen removal. Okay, so again, this just shows that the long stamens get more pollen removed with experimental manipulation. And if we measure anther separation as the distance between, as a quantitative trait, the distance between the 2 anthers, not whether or not there's 4 and 2, but how different are they? The more separated they are, the less pollen removed per visit. So the more, well, I just said the more different they are, that reduces pollen too. So why do this, right? This doesn't sound good. Well, I'll back up. We did, one obvious hypothesis was it's actually, the short stamens are to reduce the rate of pollen removal so that the flower isn't depleted so quickly. And James Thompson and Lawrence Harder had models about this a long time ago. We did a bunch of work and we can't support that hypothesis. Our current hypothesis, and my current student Robin Waterman is doing video recordings of visits. We think that maybe just having the 2 especially causes the smaller pollinators, like those small bees, to move back and forth between the anthers, and because they will feed on the short stamens, and that causes better contact with the stigma, that movement around the flower. But we think we don't have solid evidence yet, but it's looking like that. Okay, so this is the function of the stamens, that is getting pollen on pollinators. What about natural selection on anther separation? So here's the old name. We used to call it anther dimorphism, but this is again just the difference in the heights of the 2. This is standardized so that the mean is 0, but that's really about 2 millimeters of difference. And what we see here in the first year, a really long time ago, people here weren't born, right? Yeah. In 1991, we see really strong stabilizing selection. Okay, so what that would do is maintain the current mean difference between the two of about 2 millimeters. So this is, I mean, strong examples of stabilizing selection are pretty rare. Now, I used to have this thing called Connor's law of preliminary results, and if you got a really nice, clear result that fit what you knew, fit your hypothesis, for God's sake, you just stop collecting data. But I created this law back then in the early 90s because we repeated it 2 more times. Oh, I just said that this is consistent with selection, maintaining that anther separation. The next 2 years, there was stabilizing selection, but it was pretty weak, and I don't think it was statistically significant. It's a little messed up, but it wasn't statistically significant in either year. So that might mean that there isn't really stabilizing selection. It does mean, I've said this several times today in discussions, every year in the field is different. Certainly that was true in this case. But another possibility is that there's just a lack of variance for an adaptive trait. And any trait that we think is an adaptation means that it's been through selection in the past, and so less fit variants have been removed from the population. Sharon, this was even before you were in the lab, wasn't it? Oh boy. Sharon Kessler was in my lab as an undergrad, which I forgot about. I said, how did I know you? I said, don't I know you? Or something like that. So selection removes variance, and that means when we try to look for the relationship between variation in the trait and fitness, which is natural selection, we may not be able to observe it anymore. So what we did was artificial selection on the, I said before, we did this on anther exertion as well, but we did it on the separation between the anthers. And what we said, in just 5 generations, we selected just in one direction to try to get rid of the anther separation back to the ancestral condition of 6 stamens of equal height. We didn't make it all the way there, but it was only 5 generations. And these two, the lengths of the 2 stamens or filaments are genetically correlated at 0.95 or something, and that's due to pleiotropy or very tight linkage. It doesn't cause a strong constraint. And it's not just our work that has shown this. So we've got this great separation between the random mated controls and the selected lines in just 5 generations. And then I love these artificial selection studies we've done because usually they're grown in the greenhouse and then often the students will put the flowers in water and tubes and bring them in the lab, and I can look at the rack of tubes after a few generations and I know what, it's not blind, these measurements. So here's the high line, so the anther sticking, I'm sorry, this is not anther exertion, this is separation. You can see they're very separated. The top of the short stamen anther is not even quite reaching the level of the long, whereas here, I gotta look over here, you can see they're broadly overlapping in height. So here's a short one where the top of the short is about more than halfway up the long anther. So we made these dramatic phenotypic differences in just 5 generations. Okay, so then what we really wanted to do with this, though, where I started with this, I think I said this, there's no strong constraint maintaining tetradynamy across the brassicaceae, well, in wild radish. That's all I can say, because that's what we worked on. So we increased the range pretty much symmetrically and more than doubled variance. Now we can take those out to the field, and now we have lots more variance to measure natural selection on. So these are plants, and we use these potted arrays because we wanted to measure lifetime male fitness. Am I getting some feedback here? Are we okay? Oh, good, okay. I'm getting feedback. And so here people doing phenotypic measurements, pollinator measurements, and so what happens, remember, we're looking for more expanded variance so we can really see that stabilizing selection. It's not upside down. So what we found was really strong disruptive selection. Now, good demonstrations of disruptive selection are even rarer, so that's cool. But we don't understand this. And this disruptive selection on the difference in lengths of the two stamens is the same thing as what we call negative correlational selection that would act to reduce the correlation between the two, and the correlation is really high. So we know that this was a funny field, or I'm pretty sure this was just some weird year in the field, but it's still pretty cool. So opposite, and again, the preliminary results thing, I mean, we spent years doing artificial selection. It's crazy, just stop. No. So here's the two main results. 1 year of strong stabilizing selection based on natural variation, and 1 year of disruptive selection based on expanded variation. So what's still true that both of these maintain the current mean, okay? Because there was not directional selection in either year, and so even though it's disruptive, that'll create more variation in the anther separation, but it won't change the mean. And the stabilizing selection reduces the variation around, again, that same mean. And I said, we're still looking for the biological function. And the selection does depend on visitation rate, but not in the way that we would expect from that reduction of pollen removal hypothesis that I talked about. And again, there's not an absolute constraint on this trait because we can select on it and it can move rapidly. And lots of people have selected against genetically correlated traits and found rapid responses. So this idea that I sure had that genetic correlations cause these strong constraints is probably not usually true. It's probably in my book too. Well, there's Robin. So she's there just because I talked about she's analyzing video data. This was work that she did with RU undergraduates in the lab for a couple summers. So there's 4 long and 2 short stamens, and there's wild radish. I already said this. Oh, it is maintained. So that disruptive and stabilizing selection is actually on the difference is the same thing as correlational selection on the individual traits. I can talk about that more later. But that's why Robin's next to the, she's trying to fit it, the function. And again, we think that pollinator movement might be what's going on. Okay, but now we're gonna make a common transition in the talk. So one of the most common evolutionary transitions in the angiosperm tree of life is from outcrossing to selfing. And so we started working on Arabidopsis. People for years, so some people like Chris Oakley's former postdoc advisor, Doug Shemsky, didn't want me working on weedy rash because it was non-native in this country. And other people would say, you should be working on Arabidopsis, think about all those tools you have. And so I thought, wild radish might be okay because I'm getting criticized by both sides. But then we found this cool trait in Arabidopsis, so I was won over, and oh boy, those of you that know the tools, oh boy, it's pretty cool. There are some estimates of lower rates of selfing than 95%, but most of them are really in that high range of selfing. And so you can see here that here's the short stamen. We did not do this, make this SEM, very far from the stigma. And there's some pollen on the side of the style. That's not doing much good for the plant. But you can see the long stamens are right here next to the stigma. And so we predict that the short stamens don't function well in self-pollination. And so my former student, Ann Royer, now at Scranton University, my students go to Pennsylvania for some reason, I don't know why, did this experiment, this pollen removal experiment, stamen removal experiment, where she had, I think both unmanipulated and sham manipulated controls. She removed the 2 short stamens, she removed the 4 long stamens, and she removed all of them and looked at seed set. And what we see is if you have 0 stamens, fortunately, you almost never make seeds. There are a few, I guess, accidental pollinations. She did these removals in the bud because Arabidopsis at least sometimes can self in the bud, I am told. Does anyone know that that's true? I think that's true. Maybe a little bit of function of short stamen, but certainly not significantly different from the 0. And then these two, again, there's some hint that having those two short stamens compared to only having the four long, but not statistically significant, okay? So certainly there's not much function, if there is any function, of the short stamens in selfing. So it's a pretty cool trait. The long stamens are almost never lost. I mean, even in wild radish, we'll see rare, screwed up flowers with missing stamens, but almost never are the long stamens. But the short stamens, when they're lost, they're just lost, and they can be lost. So here's the no loss. Here's one with just one short stamen, and here's one that's lost both. And these can occur on the same plant. Okay, and so we basically study this as average number of stamens per plant. We tend to measure our standard set of 9 flowers spaced across the flowering range of the plant, so we're not just looking at early or late flowers. And sometimes there's a trend in stamen number that way, and sometimes there's not. We were just talking about that in the lab meeting the other day. So it's a really pretty interesting trait. And so we had an undergrad in the lab. We have a lot of undergrads in the lab in the summertime. We're a field station, not during the year. And so here's Kola George. And what he did, instead of doing experimental removals, he said, let's take advantage of the natural variation, the 0, 1, and 2, that you can find on the same plant, and score the stamen number non-destructively. So there he's doing that, and then score seed set. And so let's look at the natural variation, and again, within plants, and there's really no difference at all. There's slightly more seed set by having one short stamen, but this is obviously very far from significant. I think there's really, looks like no difference. So it's even clearer than the experimental manipulation, maybe because the flowers get a little mucked up if you go into the buds and try to remove stamens. So if we lose function, we tend to find trait loss, okay? And the best known examples, like cavefish, loss seems to be driven rapidly by selection. That's my reading of the cavefish literature. That seems to be where they're coming out. But here, the selection, I'm thinking, is likely weak. It's loss function, but they don't cost much. There's not much tissue there, although we're working on that. I'll show you some data. Maybe we've caught evolution in the act. Maybe it's ongoing, and we can study those mechanisms. The fact that there's still that variation from the ancestral 2 short stamens suggests we've caught it in the act, or maybe there's some kind of balancing mechanism that's maintaining this variation. So, oh, they're not supposed to be there yet. There's Anne again. Another thing she found was she looked at accessions from across the European range and found a decline. So the stamen loss is rare in Scandinavia, but it's much more common in the Mediterranean across the native range. Okay, so there's this latitudinal decline. And now we can see why Sam Perez and Sophie Bicey are there. Well, not yet. This is a growth chain common garden, so these are genetic differences depicted, hopefully now. No, yeah, I already said that. Okay, there we go. I'm usually good at animation, but not this time. So what Sam and Sophie did, and Chris Oakley, Sam was in Doug Shemsky's lab, my student, but hanging out with Doug. He found a parallel altitude decline in the Pyrenees in Spain. So Pyrenees, low latitude, so at low altitude, there's the stamen loss. But once you get to about 1,000 meters or so, you pretty much have 2 short stamens. There's still some variation, but it's a pretty strong decline. So, well, I'll back up. So there are, well, I've been saying there's lots of traits that show parallel latitude and altitude declines. There are some, but we haven't found that many. So if you have good examples, but because high altitude and latitude are similar kind of environments, shorter growing season, obviously colder winters and things like that. So there are some like that, but we can't imagine short stamens necessarily would be one of those, or we can't understand why. If the short stamens are there for pollinators, for the rare outcrossing events, you might think it should be at low altitude. And certainly in Spain, our collaborator Javier Picot, there's way more pollination going on. Pollinators visiting, not a lot because it's Arabidopsis, at low altitude, not way up high in the Pyrenees. So it's almost backwards if the short stamens are there for outcrossing. Our evidence of selection in wild radish says they do function in outcrossing because there's selection on the distance between those two. Okay, so what we're doing now, is this what we're doing now? Why is that? Okay, is doing work across both those clines, both the latitudinal and altitudinal extremes. We're starting in Spain, and so this is work that I should have Jimmy Bingman and Ava Garrison's pictures here, but they're not there, sorry. I think I just forgot. So this is repeating Ann's, let's just put this, so there's Ann's figure that I just showed you, repeating it at both high and low elevation, because one hypothesis is that the short stamens, even in selfing, have more function at high altitude or latitude, and probably not. So at the low site, it's quite clear that the 0 and the 2 short stamen plants have the same seed set. Boy, that yellow doesn't show up too well for me. There's a yellow, can you see that? Here's 4 and, shoot, I forgot. And this is the sham operated, is the yellow. Here's the unmanipulated, so we're screwing the plants up, they're screwing the flowers up, they don't set as many seeds. And essentially the same pattern at high altitude, although there is this interesting hint again, a little bit like Ann's hint here, that maybe 2 short stamens at high altitude anyway give you a little bit of self-seed set. But as Jimmy points out, well, if you only have 4, it doesn't matter. So Jimmy's point, well, what does this even mean in terms of in nature? Because they always have the 4. So we're still working on this. We're going to repeat this again next year, so stay tuned. But not a lot of evidence for short stamen function in selfing. And then this is a repeat of Kola George's experiment, but in low and high, using the natural variation in short stamen number scored non-destructively, count seed set, and just like Kola's results, there's just no difference at all. I like the experiment also, but in some ways this seems a little more convincing because we're not screwing up the flowers. Oh, and yes, there's Kola's graph for comparison. Okay, so what's going on here? Why do we have these parallel clines? It doesn't seem to be a difference in function in selfing, which I've just shown you. So there could be selected. We have 3 hypotheses that we're testing. Selection to either maintain the short stamens at high or to lose them at low, maybe to reduce costs of producing them. There could be just loss through drift. So the standard trait loss with relaxed selection, I don't love that term, but when selection is reduced, as in the case of selfing or Arabidopsis, where the short stamens have lost function, the classic is then the trait's lost through drift. But we have an alternative hypothesis because we know that natural variation in genomic variation is less at high altitude and high latitude due to, we think, genetic drift causing through founder effects as the populations re-migrated up slope and up north after glaciation, they lost genetic variation. And several studies have shown that there's a pretty clear cline in just SNP variation from south to north and low to high elevation. So it might be that since they have not lost them here where they have less variation, maybe there is selection to lose them, but there just isn't enough genetic variation in short stamen number for that to happen. So we have 2 separate opposite drift hypotheses, which is sort of confusing. There's what I just said. Okay, there is more drift. And then our last is pleiotropy, because there are other traits, not as many as we thought, but there definitely are other traits that show these clines that are adaptations to short growing seasons at high latitude and altitude, and so maybe there's just pleiotropy, and maybe the short stamens are close to neutral. They don't cost much and they don't do much or anything, so maybe they're just being pulled along by genetic correlations caused by pleiotropy. So we're trying to look at all these, definitely not mutually exclusive. Maybe the two drift ones are, I don't even know. Could they both be going on at the same time? I guess they could. Certainly the effective population size is higher in the south, so there's less likely for drift to cause anything, but anyway, it still could happen. So here's sort of a look at selection, and there's Chris's name there. So what if you don't know about this work, you should. Chris has been involved with Univ and Doug Chemsky and others in an incredible study looking at local adaptation in Sweden and Italy. And this is old. You have 8 years now, right, Chris? I only have 6 here. And so what I did, I actually can't, so they measured fitness. I'm backing up. They made recombinant inbred lines. I think that's coming later in the talk, it is, sorry. Between Sweden and Italy. And they were looking at these traits down here, trichome density, freezing tolerance, flowering time, and some others. And what Ann Royer was in Doug's lab in her first year in graduate school, and she's found the stamen loss in the Italian parent. And so she used those same recombinant inbred lines and scored stamen loss, and you'll see results from that later. What I did was, well, Yoon wouldn't share the fitness data of these rills across all these years, but we've got a phenotype and they're inbred lines, so they're genetically the same. And so Yoon plugged those into a selection gradient analysis with these other traits, and what you see is it's variable. So I've highlighted the significant years. So there is selection to maintain them in Sweden. That's what the hypothesis suggested, and the mean over 6 years is significantly positive. In Italy, it's almost significantly positive, but there are some years, including one significant year, where there is selection to remove the short stamens. And something else I've said in several conversations, that every year in the field is different. Field work is necessary for evolutionary biologists, but incredibly frustrating because of this fact. And so it may be that, so the net over this 6 years, and I guess we need to do the last 2 years, because maybe there'll be lots of negative selection in Italy. No, the Connor preliminary results law would say no. Okay, we won't do it. Anyway, there is definitely sometimes selection to remove short stamens in Italy. Now, the caveat here is these are great fitness measures in the field, but we measure short stamens in the growth chamber. And so I'll talk about our plans to do this, we think, better in the field in the future. But I also want to point out that in Sweden, the selection to maintain short stamens is stronger than freezing tolerance. Not significantly so, but it's incredible. Now, I'm not sure I believe that. So maybe it's pleiotropy with a trait that's not included in this analysis, because these are these three in stamen loss this is a selection gradient analysis, so the correlations among those are corrected for, but not on measured traits. Maybe I should believe in strong selection on short stamen numbers, because the data keeps suggesting it, but I don't know where it would come from. Okay, so that selection, maybe. I mean, definitely selection to maintain them in Sweden, it looks pretty strong. Yeah, okay, I said that thing. This is why I have bullet points, I guess. I said that. I guess I'm saying it now. Okay, so what we're working on now is making nils, and Juan Diego and Chris have been super helpful here because they have lots of single nils. And so what Ava Garrison, my postdoc, is doing is crossing them. We now have double nils. We haven't tested the doubles, but I haven't told you about the QTLs yet. Oh, I messed this up. Okay, there's some QTLs, and we'll talk about nils soon, unless I forget, and then you can yell. All right. So drift is hard. And, you know, I made an argument in the grant proposal. We really don't know very much about the effects of genetic drift on phenotypic evolution, and I think it's true, and it's because it's too hard. But here's what Sophie Bicey has done with the Pyrenees populations to try to get at this. So here's the altitudinal gradient that I showed you before that she and Sam did. And then we got sequence data from other collaborators, and she showed that quite clearly, and I already said this before, that this is just pi, nucleotide diversity based on sequence diversity. It goes way down at higher altitudes, almost certainly because of founder effects due to drift. There may also be smaller population sizes currently at high elevation, and I think Xavier Picot, who actually collected these with the late Steve Tonser, I think he suggests maybe there's less gene flow as well. So all those things together would reduce pi. And then what Sophie did was, well, can we see if this is what's driving the stamen loss, right? The drift hypothesis. Is this low variance constraining the loss of short stamens at high elevation? So she did a multiple regression with both of these together, predicting short stamen number. And what you see is elevation is still there. It's that relationship's almost unchanged by including pi in the model, and there's really no relationship with pi at all. So it's not perfect, but it strongly suggests that whatever's causing the decline is not genetic diversity. These genetic diversity differences caused by drift. Okay, and we have more stuff on drift coming up. I'll mention it briefly at the end. I just said that. Okay, so what we're working on now is a pretty giant growth chamber common garden where we're using lots and lots of lines and replicated across the native range. And so, yes, so we want to look for genetic differentiation for a whole lot of traits, more than 30 traits. Some of these are just floral traits that are related, we think might be related to short stamen number. Others are traits that we know that they're declines, either latitude or altitude or both. Well, that's mostly what we're doing. Others, there's just phenology and things that are easy, although some of them show declines for sure. And so we have two things we call in the broad set using the 1001 genomes and the lots of populations, but 1001 genomes per location, per population. Often there's only one or a few lines. So that's cool for just, well, yeah, that works for differentiation, but it doesn't tell you anything about what's going on within populations. And then we have what we call the intensive set, and these are lines that Unagren and maybe Chris has helped collect some of these across Scandinavia and Italy, and then the Spanish lines that Xavier Picot and Steve Tonser collected. That's mostly, and here we have multiple lines per population. And I'll try to say this gently, they were collected by evolutionary biologists, so they're in more natural locations, so there might be less likelihood of recent introduction into those populations by human-caused gene flow, seed movement, I should say. Okay. And these are mostly at latitudinal extremes. We don't have a lot in the middle of Europe, so that gives us some power, though, to test the hypotheses, because that's the phenotypic extremes as well. I think this is where we're at for planted. A whole lot of 1,000 lines, 2,500 plants. Yes, because sometimes that doesn't work. No, it's more than that, because we plant either 3 or 4 replicate plants per line. I did some math wrong there, or somebody did. And then we measure 9 flowers per plant for stamen loss, so that's really well estimated. Most of the other traits are measured on just once per plant, but that's still pretty good. And so here's some preliminary results. Well, this isn't results, this is just telling. Okay, I'm going to present preliminary results from the first 2, but we're going to definitely plant more. We have planted more of the intensive. We may go back to the broad for more too. But so let me just give you some quick, pretty preliminary results. So here's Charlie Anker, who did a master's in my, just finished over the summer, a master's in my lab. So this gets really confusing, all this stuff. So I'm going to try to walk through it, and I may be wrong about some of this, so I'd be happy to hear about it. So here's what I'm thinking. For pleiotropy to explain it, the short stamen number declines. There needs to be another trait that also shows decline, okay? And the two need to be correlated with each other. But more than that, the trait, kind of similar to the multiple regression that Sophie did with pi and latitude, the trait needs to predict short stamen number after correcting for latitude or altitude. I think this is right, because what that means is, because they're all intercorrelated, because these are other traits that show clines, and the short stamen number shows clines. And of course, latitude and altitude aren't something that plants adapt to, right? They are proxies for lots of other environmental factors that vary along latitude and altitude. They don't know, plants don't know what latitude or altitude they're at. Okay, so she measured 18 of the traits and then regressed them on latitude. And so here's a multiple regression of these 3 traits on, oh, no, this is on latitude. I haven't gotten to this is the next slide. So what we see is there are 3 traits with pretty significant latitudinal clines, but the R-squareds aren't very high. And flowering time, as some of you surely know, is a very well-studied latitudinal cline in Arabidopsis, but it's pretty weak in this set, this subset of the 1001 genomes, whereas short stamen number is, latitude explains 23% of the variance in short stamen number. So it's a really strong cline. So that's cool. Again, the data keep telling me, yes, short stamen number is really important, but I'm still kind of skeptical. So now we'll talk about do those other 2 traits of flowering time and I forgot what it was, leaf number at bolting, rosette size, do they predict short stamen number after taking latitude out? And the answer is no, they don't. And it's not even close to significant. So there's a strong effect of latitude after you take these other 2 traits out. So I think that's suggesting to me that they're not causing the cline through pleiotropy. And I think there's a bullet about to show up. Yeah. So for these 18 traits so far, I don't think we have good evidence for pleiotropy. I'll show you some data later if I start going much faster. We're still going to look at flowering time anyway. There's other possible reasons to expect that. So now we have the other set, the intensive set, and now I'm going to go straight to that multiple regression. And what we see here is now we have, now we have altitude. We have a better set of altitude. There wasn't enough altitude variation in the other set. Very strong clines. Flowering time, again, is not there. So flowering time, so again, this is regressing short stamen number on these 4 traits, multiple regression together, correcting the correlations among these. But here's this trait flowering rate, and Jimmy Bingman, my student, first just kind of noticed this in the fields, and that's why we started measuring it more precisely. And so plants that flower more rapidly have fewer short stamens. So again, maybe the cost saving of those teeny tiny short stamens are allowing the plants to produce flowers more quickly. And we have some evidence that is related to, flowering rate is related positively to fitness. If you flower more quickly, you have higher lifetime seed set in the growth chamber so far. We'll do it in the field soon. Okay, so maybe flowering rate is a candidate for pleiotropy. It's not a super strong relationship. I just said that. So kind of to back up for a second, in wild radish, tetradynamy has an unknown function, but there's strong selection on it, at least 2 of the 4 years that we looked at, especially when we expanded the variation. Short stamen has lost most or all of its function based on the functional studies I showed you. So this selection there's not selection to maintain them. And so the argument in the grant proposal is weak selection adaptation is more likely to be constrained by drift pleiotropy. And I've thrown in epistasis because we found some epistasis. So I've spent my career doing quantitative genetics and estimating G matrices, genetic variances and covariances, genetic correlations, but they haven't helped so much with constraint and these kind of questions, well, constraint in particular, and single locus population genetic theory for drift versus selection are very well developed. Remember, I said drift is hard. And so this is a case where, And for maybe none of you in this room are trying to find genes underlying quantitative variation controversial. It is a little controversial in the evolutionary community. I think partly because we don't have a lot of results to show for it. Chris Oakley does, not a lot of other people. Anyway, so this is the argument that I made in the proposal, that we really do need to get to single genes. And I've made this argument more broadly in other contexts. But so here's what you needed to see before. So here's Anne's latitudinal cline, and there's Rhodus in Sweden, and there's Castelnuovo, Italy, the lines that did you do the initial crosses, Chris? No, okay. Did you do the initial crosses between them? You're not that. Somebody asked me at lunch whether we were postdocs together. And I said, don't tell Chris you asked that question. I was a full professor probably already. I think I was. Anyway, when you were a postdoc. So they some older people crossed them. No, they're probably younger people in Doug's lab, I'm guessing. I don't know. Anyway, they were crossed. They made these nice rails, and they genotyped them. And then, as I said, when I got ahead of myself, Ann Royer spent her first year in Doug's lab, and she scored stamen number on them. And so this allowed her to do this QTL mapping. And she found a whopping big gene at the beginning of chromosome 5, a pretty big one on chromosome 3, and a pretty small one on chromosome 1. So she had good power with these rails. These rails they're an incredible resource, really. The tools in Arabidopsis. You can't even make recombinant inbred lines in radish, because if you try to inbreed them, we've done this, inbreeding depression is so strong that they just can't make it. And they don't make normal phenotypes anyway. So Anne found this, and there's quite a bit of epistasis between the chromosome 5 and the other two, the QTL chromosome 5 and the other two. So strong effect. And so here's maybe where I'll say here's where Ava, not Anne, Ava is making the double and triple near isogenic lines where she's introgressing the Italy portions of these chromosomes into the Sweden background and vice versa, Sweden into Italy. And then we'll take those when they're done and put them, the single, the double, and the triple nils that have all three introgressed both directions, and take them out in the field and measure fitness in Sweden and Italy. And the idea was, actually only the Italian background will go in Italy and the Sweden background will go in Sweden, because Chris and Doug and Yoon have shown strong local adaptation between those two. We're not interested in that. Again, I'm hating on short stamens again, but I don't think the effects are going to be that strong of short stamens, so we really want to isolate the effects of short stamens and have the rest of the genome locally adapted where we put these out. Okay, I better start wrapping up. So here's the same slide again. And so there is selection on these other traits, and probably more, I don't even know. But so that's another element, another way to think about it. So before I said they need to have a trait, for a trait to cause the stamen loss cline through pleiotropy, it has to also show a cline. Well, that cline has to be the cline would probably be created by selection. So here's some other traits that we should look at, and we're mostly not. Well, I won't talk about that right now. But and so here's these other traits that Chris and other people associated with the Shemsky and Ogren labs, and there are on chromosome 1 and 3 there are overlapping QTL. These are broad regions. They're not necessarily that close, but there's flowering time, fitness, differences in ovule number, and again, flowering time, freezing tolerance, and fitness. We're definitely not doing freezing tolerance because I don't have the chambers to freeze things, and that's too hard. But flowering time, we definitely are looking at, even though, remember, the earlier results based on the correlations flowering time does not predict stamen loss after you take latitude or altitude into account. That's just either latitude or altitude into account. Okay. And then here's Madison and Sam Perez. They found in a different rill set, Sue and Cass. So Cass shows stamen loss, and also there's rills available. There's overlap. These are a little funky, but I haven't shown the overlap yet. There's one on chromosome 4, and there's one or two on chromosome 1, and another strong one on chromosome 5. Oh yeah, they measured lots of stuff. And there is a fair amount of overlap on 1 and 5. And so what Madison's doing right now, Madison's not my student, she's David Lowry's student, but she did a rotation in my lab and she's finishing this up, searching for these parallel substitutions. Oh boy, I'm on automatic. Did I push some button here? It's okay, I don't have much time left. I'll talk really fast. She found 35 shared SNPs. 25 and MIB29, there's a strong candidate gene. She ordered those knockouts and some others. Nope, no phenotype, no stamen loss. But she just ordered knockouts for 6 more lines. How do I turn this off? Did I push? It's okay. I'll just push backwards. Right, so we're hoping to, maybe we'll get lucky with one of these knockouts. Madison's so thorough, Chris, I don't know if you're interested in this, she had all 4 parents resequenced because they were different sequencing quality for these 2 rill sets, so she has really nice sequence data. Okay, so wrapping up, direct selection, I showed you there's selection to maintain short stamens in Sweden and Italy overall, but there were some cases of selection to reduce short stamen number, negative selection in Italy. And I showed you lack of function at high and low altitude in Spain by both the natural variation and the experimental removal. We're going to measure fitness in Spain this next year. I didn't do it the first year because, again, I didn't think there'd be a big result, but no, I'm not so sure. So why not measure fitness? It's not that hard in Arabidopsis. Another key thing of Arabidopsis, I spent a lot of my career yelling at plant biologists to measure male fitness because they don't, and doing lots of molecular work to do paternity analysis. That's what the wild radish results were based on. Arabidopsis, it's mostly cell thing. If you count seeds, annual plant, that's almost all the fitness that plant has, male and female. You haven't got all the male fitness because there's a little bit of outcrossing, but it's an incredibly good fitness measure, and evolutionary biologists suck at measuring fitness. This is a problem with our field. I showed you the evidence against constraint due to drift, constraint to loss at high altitude, because if you put pi in the model, that doesn't explain short stamen number at all. But we want to estimate broad sense genetic variation within these intensive lines where we have a number of lines per population. Is there genetic variation for selection to act upon in those or not? Pi is cool, but genomic measures of genetic variation don't usually correlate very well with quantitative genetic measures of variation of specific traits. And so we want to do that. And I have a colleague, Gideon Bradford, who does all kinds of crazy genomic analyses to understand selection across, using genomic tools genomic data to understand selection and drift across space. And then finally, maybe flowering rate. We can also estimate the genetic correlation within these intensive lines. And we're doing work to find the genes. So I'll just finish there because I am way out of time. There's my pictures of my lab and names and other collaborators, and I cut off, that's funding from NSF, not funding. I'd be happy to take questions. Thanks.
One quick question, and then whoever wants to stay, whoever would like to take any answer. Go ahead.
Who did you work for at Purdue?
Dr. Dick Doherty. I think he went by Richard in food science, and I studied tomato juice consistency. I was the last person to come. It's a long story, and I think, anyway, I'll just.
That was a short answer.
That was a short answer. Yeah.
Okay. I had a question because while you're describing your work, I was
thinking on a developmental perspective, what would be the function of the short stamens? So did you ever looked at the elongation time of the stamens in proportion to the carpel? Because I would imagine that on a flower that is maybe growing on a colder environment, the elongation of the carpel might be delayed, and so those short stamens could function to assure pollination.
So I think what you're saying is, I mean, that would all be in the bud, because that's all done when the flower opens. I've studied this carefully in wild radish. The pistil still elongates a little bit, and then it goes crazy if it's pollinated in both species, because that's the ovary expanding. But the stamens are done well before a bud opening. Elliot Byrowitz's lab looked at that. So, but
more or less with the same size on a closed bud, and then I have that feeling, I don't know, that the longer stamens elongates and the short continue short. So if you look at in terms of flower development, is the carpel, the pistil, elongating slower than the short stamens would have a function.
well, maybe, except I think, do you have something to add here? Or, oh, okay, sorry. You seemed enthusiastic, which is great. So anthesis, the release of the pollen, occurs quite late. And I said this thing about does pollination, I always was told pollination occurs in the bud in Arabidopsis, but I just suddenly realized I don't know that that's true. But I think it does at least sometimes occur, but it's late. I'll talk to Ava and Jimmy, because they've opened a whole lot of buds to do these stamen removals and things. Or Ava's done emasculations to do crosses. I think the short stamen is well below the stigma even there, but that's possible. So the short stamens actually initiate slightly later, 2 floral stages later. I can't remember, like 10 versus 12 or something in the however many floral stages of development. And so, yeah, I think they're always behind, if that's what you were saying. They're always shorter because they start later. Does that help? Or?
So I was thinking if, I don't know if you have data on the elongation of the pistil itself and how it correlates with the stamen elongation. I would imagine that the flower growing on lower temperatures would take more time. Maybe there was some delay. So I was...
Okay. Do you think the pistil would be delayed more than the stamens? I mean, that's what you're saying is they're close. I mean, again, when the pollen's released, I think that's really late. Well, you're saying could it still be very short and down near the shorts? Could the stamen be down where the short stamens are and colder? Well, we should look at that. Yeah. Okay. Cool.
Okay, let's thank one last time to Dr. Conner. If whoever has any more questions, just reach him personally.
I'll just say.